Good morning, everyone. I am delighted to welcome you all today. Thank you for being here. Today, I will be talking about technology transfer for analytical method. We will explore how analytical methods are transferred from development to practical application, ensuring quality and consistency. Over the course of this presentation, we'll cover key concepts, best practices, and real-world examples. Let's get started and dive into this important topic. Let's take a moment to understand what analytical method transfer, or AMT, actually is. It's a documented process designed to ensure that when we transfer an analytical method from one location to another, specifically from the sending unit to the receiving unit, it performs equally well. The goal here is to make sure the method works just as effectively at the new site as it did originally. This is crucial because it helps us demonstrate that different laboratories can produce consistent and reliable results. Now, why do we focus on this? Well, the main objectives of AMT are to ensure that the method is accurate, precise, and reproducible. These are the cornerstones of reliable data. We also want to demonstrate that the method is suitable for use at the receiving unit, which means it meets all necessary performance criteria. Another key point is maintaining data integrity and ensuring compliance. This is vital because the data generated needs to be trustworthy, especially when it's used for regulatory submissions. Ultimately, AMT is about making sure that the transfer process supports high-quality, compliant, and reproducible analytical results across different laboratories, reinforcing confidence in our data and processes. Now, let's take a closer look at the method transfer lifecycle. This process begins with transfer planning, where we outline the scope and objectives of the transfer. Next comes protocol preparation, which involves developing detailed procedures to ensure consistency. Training is then conducted to make sure everyone understands the methods and expectations. Once the team is prepared, we move on to execution, where the actual transfer activities take place. Afterward, data evaluation is critical. We analyze the results to verify that the transfer has been successful. Finally, we compile a transfer report that documents all the relevant information and outcomes. When it comes to the types of method transfer, there are several approaches to consider. Comparative testing involves direct comparison between the original and transferred methods, like comparing HPLC assays between the success and receiving units. Co-validation is used when the method is transferred between labs or teams to confirm consistency. Revalidation may be necessary if the method parameters change significantly, ensuring ongoing reliability. Verification is another option, often used for routine checks or smaller adjustments. It's important to remember that the choice of transfer type depends largely on the risk involved and the complexity of the method. For example, a straightforward HPLC assay might just require comparative testing, whereas more complex methods could need co-validation or revalidation to ensure quality and compliance. Let's talk about the transfer protocol, which is a key regulatory requirement we need to understand. First, the protocol itself must include several essential components. These are the objective and scope, which clearly define what we're aiming to achieve and the boundaries of the transfer. Responsibilities are also outlined, specifying who is responsible on both the submitting unit, or SU, and the receiving unit, or RU. Materials and methods are documented to ensure everyone understands the procedures and resources involved. The protocol also covers the experimental design, which is crucial for ensuring consistency and reliability. Acceptance criteria are set to determine whether the transfer has been successful, and procedures for handling deviations are included to manage any unexpected issues that might arise. It's important to remember that this protocol is mandatory before any testing or transfer activities begin. Now, regarding the scope of the methods transferred, it includes several key assays. For example, API potency testing, impurity profiling, dissolution testing, residual solvents analysis via gas chromatography, and microbiological methods. These methods are vital because they cover testing of not just the API itself, but also intermediates and cleaning samples. Overall, ensuring that these protocols and methods are well documented and followed guarantees the quality and consistency of our products throughout the transfer process. Let's take a moment to discuss the prerequisites for a successful transfer. First and foremost, the equipment involved, such as HPLC, GC, and others, must be qualified. This means they've been validated and are functioning properly. Equally important are trained analysts who understand how to operate this equipment correctly. We also need reference standards, which are critical for ensuring accuracy and consistency, and approved standard operating procedures, or SOPs, that guide every step of the process. It's essential that both the equipment and personnel are properly qualified and trained before starting the transfer. This helps maintain the integrity of the process and results. Now let's look at the roles and responsibilities. The sending unit, or SU, has several key tasks. They need to provide a validated method and supporting reports, and also conduct training sessions for the receiving unit, or RU. Additionally, they must define clear acceptance criteria up front, so everyone knows what successful transfer looks like. On the other hand, the receiving unit's responsibilities include verifying the method's performance, making sure the equipment is ready and in good condition, and executing the protocol as outlined. Finally, it's crucial that both parties, SU and RU, approve the results and reports. This mutual agreement confirms that the transfer was successful and that the data is reliable, aligning with the overall goal of ensuring quality and consistency in our processes. Now let's take a closer look at the acceptance criteria. These are the standards we need to meet to ensure our method is reliable. First, we have the %RSD, which measures precision, how consistent our results are across multiple runs. Then there's %recovery, which assesses accuracy, how close our measured values are to the true value. We also evaluate the bias between the system under test, or SU, and the reference, or RU, to identify any systematic difference. Lastly, system suitability tests help us confirm the system's performance before we proceed. It's important to remember that these acceptance criteria must be predefined and justified, meaning we establish them beforehand and explain why they are appropriate for our analysis. Regarding the experimental design, a typical approach, especially for assay transfer, involves using two labs, our SU and RU, working together. We have two analysts performing the tests to account for variability, and they analyze 3 batches of the sample. Each batch is analyzed in triplicate to ensure precision. Once we gather the results, we compare them statistically to demonstrate that the methods are equivalent. This process helps us confirm that our transfer is successful, maintaining accuracy and precision across different labs and analysts. It's all about building confidence that the method will perform consistently in real-world applications. Let's now look at the statistical evaluation methods we use to ensure our data is reliable. First, we have the t-test and f-test, which are fundamental tools for comparing groups and checking for differences or variances. These tests help us determine if observed differences are statistically significant or just due to random variation. Next, there's equivalence testing. This is particularly important when we want to confirm that 2 methods or labs produce results that are essentially the same within a predefined margin, like an assay difference of less than or equal to plus or minus 2%. Finally, confidence intervals give us a range of values within which we expect the true parameter to fall, with a certain level of confidence. These intervals help us understand the precision of our estimates and whether our results meet the acceptable statistical limits. Overall, these tools allow us to ensure that our results are not only accurate, but also consistent and within acceptable limits, supporting the integrity of the entire process. Let's take a moment to look at some of the common challenges we face in our processes. One of the key issues is instrument differences, which can lead to variability in our results. This is something we need to be aware of and manage carefully. Another challenge is analyst variability, meaning different people might get slightly different results even when testing the same sample. This highlights the importance of standardizing procedures and training. We also encounter method robustness issues, where our analytical methods may not perform consistently under different conditions, risking the reliability of our data. To address these challenges, we adopt a risk-based approach guided by ICH-Q9, which helps us prioritize and manage potential risks effectively. We also conduct robustness testing to ensure our methods can withstand various conditions without losing accuracy. Proper training is another critical component. Ensuring that everyone involved is well-trained helps reduce variability and improves overall reliability. By combining these strategies, we can mitigate risks and maintain the integrity of our analytical processes, supporting our overall goal of quality and consistency. Let's take a look at this case study to see how we approached a common challenge in HPLC analysis. The method involved analyzing related substances, which can often be tricky when it comes to peak resolution. The issue we faced was a peak resolution failure at a specific retention unit, RU, which meant that the peaks were not separating clearly enough to be accurately identified and quantified. The root cause turned out to be variability in the chromatography column itself. Variability can stem from many factors, such as column age, manufacturing differences, or even improper handling, and it can significantly impact the separation quality. Our solution was to adjust the mobile phase composition, which is a common and effective way to improve peak resolution. By fine-tuning the solvent ratios, we aim to enhance the separation of the related substances. After making those adjustments, we re-verified the method to ensure that the changes improved the resolution without compromising other aspects of the analysis. The outcome was very positive. We achieved a successful transfer of the method with results that were fully compliant with our specifications. This case really highlights the importance of understanding the root cause of issues and applying targeted solutions, which ultimately supports the overall theme of ensuring robust and reliable analytical methods. Let's wrap up with the conclusion. AMT, or Analytical Method Transfer, plays a crucial role in ensuring data reliability across different sites. This consistency is vital for maintaining quality and confidence in our results. To achieve this, it requires a strong protocol, something clear, detailed, and well-structured to guide the entire process. Along with that, we need robust statistical evaluation to verify that data from various sites is comparable and accurate. And of course, regulatory compliance is essential. Meeting these standards not only ensures the integrity of our data, but is also critical for gaining GMP, or Good Manufacturing Practice, approval and regulatory authorization. In the end, these elements, strong protocols, solid statistical evaluation, and regulatory adherence, are what make AMT a reliable foundation for successful drug development and approval. Thank you. If you need any further support, please don't hesitate to reach out to us. We're here to help and ensure you have all the resources you need. Thank you so much for your time and attention today. We appreciate your interest and engagement. Feel free to contact us with any questions or for additional assistance. Thanks again, and have a great day.